Hi, everybody. Hope you're having a great week. I'm sure I'm having a great week in Costa Rica. Can't wait to tell you guys about it when I get back. But I want to go over chromosomal mutations with you guys today. So if everybody can make sure they get out this chromosomal mutations packet, get a pencil, make sure you're ready to go because I'm going to go through explaining chromosomal mutations, and then I'm also going to stop and give you a chance to make sure that you have things written down that you need to have written down so this is completed. Okay, remember, everything you're doing is going in your binder, and when I come back, I am going to be checking that all your work is complete in your binder to give you your other grade, which is your participation points for me being gone. So make sure you're getting everything kept up with, okay? Okay, so chromosomal mutations, here we go. So in our last unit, we learned about specific types of gene mutations, right? We learned about if a nucleotide, like one of the A's, T's, or G's, or C's, something was changed about them, and then that affected the amino acid, which would affect the type of protein. So if you remember, we could have two letters switch places, and that would be substitution mutation. We could have deletion, where one of those nucleotides, the letters, got pulled out, or we could have insertion, where one got put in, right? So those were our types of gene mutations, and it would mess up one protein because of an amino acid change. But in a chromosomal mutation, so if we look on here, a chromosomal mutation is talking about the entire chromosome. Okay, and in a chromosomal mutation, there's a change that's done to a part or a piece of the chromosome. And remember, the chromosomes are just the DNA coiled up, right? So if we look at the DNA kind of uncoiled, remember a gene is just a part of that chromosome. Okay, so in a gene mutation, it just affected one gene and the protein it made. But if we're talking about a chromosome mutation, then it's going to affect many genes. So it's going to have a much larger effect, which is what this slide is going to show us. So in a chromosomal mutation, it's going to be more severe, which means it's worse. Okay, so it's going to be worse because it's going to affect the entire section of the chromosome, which is many genes. Okay, so on your packet, go ahead and look at that. Let's make sure we have this first one answered. It says gene mutations are usually less severe than chromosomal mutations. And this shows us that chromosomal mutations are more severe. So this one says gene mutations are usually less severe. Is that going to be true or false? Yes, that's going to be true. So for your explanation, right, you can explain that a gene mutation only affects one protein, where a chromosome mutation is going to affect many genes, so it's going to affect many proteins. Okay, so gene mutation only affects one protein that's getting made. Chromosomal mutation is going to affect many genes, which will affect many proteins that get made. And then the next section says mutations always hurt the organism. And in this, the word always makes me think it's probably not correct, because in science, there's very rarely something that is always the case. Okay, so and we're going to learn in further on in the slides, there's actually sometimes when a chromosomal mutation might be helpful. So it does not always hurt the organism. So you can go ahead and check false for that one. And our explanation could be that there could be positives that could come from a chromosome mutation. Okay, so you should have this first box completed now. Now we're going to go through a slide and explain each one of these in this box, but we're going to get to the end and we're going to have this box to copy down. So all you have to do is pay attention to my explanations in the slides right now. So first one is deletion, and that one makes perfect sense, right? We already had a deletion in a gene mutation when one nucleotide was taken out. This type of deletion means we're going to be deleting an entire section of the chromosome. So this piece is taken out, it's just lost, and that chromosome goes back together, but now it's missing a whole section and it's smaller than it was before. So that's deletion. Another one is translocation. Okay, and if we think about the word location, that means that a gene is going to switch its location. It's going to move to another part. Okay, so it's going to relocate from one section to another section of the chromosome. Okay, so translocation. And then we have duplication. Duplicate something means to copy it, right? So if we look at this little part of the chromosome represented by the orange here, so a piece of that is going to make a copy, and then both of those pieces are then going to be in the other chromosome. And this happens, like it says, between homologous chromosomes. Remember, homo means same, so it is the same chromosome. Okay, but this would be like from the mom and this would be from the dad. And when it makes a copy, we get a copy of this extra piece here. So that's called a duplication. And then we have inversion. So if you invert something, it's flipping. Okay, and I think on our last test, this word was in one of those as an example, and some people were getting it confused with substitution. Right, which kind of makes sense. But substitution happens with the genes, like when an A gets taken out and a T gets put in. Okay, in inversion, nothing gets taken out. All it does is get flip-flopped, right? So we've got, for example, this section represented by the yellow, this section represented by the blue, and they just switch places. So the blue's now on top and the yellow's on the bottom. So that's inversion. They're inverting their position. Okay, so here is, which I know my video is kind of taking up part of your space there. So here is the section that you now have in your packet. Okay, so you have the pictures of each one happening, which I think are really great pictures because I think it shows really well what's going on in each one of those examples. But I would like you to go ahead and copy in the top of this. So for example, where it says deletion, when a piece of the chromosome breaks off and is missing. So I'm going to go ahead and wait about a minute and I'll let you guys get that copied down because you definitely need to know all four of these. Hey, everybody, make sure you're doing your work. Okay, no side conversations. Everybody get these copied down, please. Everybody about finished up? Make sure you have all of those copied down so you can make sure you have those to study from. Okay, once you have those copied down, go ahead and switch to the next page because we are going to talk about another situation that is known as nondisjunction next. Okay, next page. Okay, so nondisjunction. So the explanation that's written on here is kind of in fancy words. So let's just talk about it. You guys asked me about this, and I think just about every class, right? When we were talking about how humans have 46 chromosomes and how you have to have 23 from your mom and 23 from your dad. And you're like, what if that isn't the case? What if you have more than that? What if you have less than that? Okay, and what that's called is nondisjunction. Nondisjunction is when the chromosomes line up in the middle in their homologous pairs, right? So we're talking about during meiosis. So during meiosis 1, when those chromosomes pull apart, they're supposed to pull apart and have half go to one side and half go to the other side. However, in non-disjunction, when they pull apart, it would be as if I could pull one of my fingers off, right? If I was using this as an example, and then there would be an extra on this side and there would be one missing on this side. So that's non-disjunction. When the chromosomes pull apart to go to the other sides during meiosis 1 and an extra one gets pulled over, or sometimes it's just even a piece of one gets pulled over, okay? And when that happens, you get too many in one side and too few in another. Two of the words that we commonly use is each little set should be, right? We start out with our 2 and then we should end up with 4 chromosomes. Let me start over. So we start out with our 23 pairs of chromosomes, right? And then during meiosis, they make their copies, they divide, and then they divide again, right? So we should have a set of 23 in each one, okay? So each one should be one part of a pair, okay? So we should have 2 in each. But if we have an extra of each pair of chromosomes, then an extra 2 would go to 3, so it would be 3. So like a tricycle has 3 wheels, right? Triangle has 3 sides. So trisomy is when there is an extra chromosome, okay? Trisomy, there's an extra chromosome. If you are missing one, so you go from your pair to just having one, then that's called monosomy. Mono means 1, okay? So let's look at what that could mean. Oh, actually, let's go ahead and make sure you have a chance to write down the definition for non-disjunction. I just explained it. So on this next page, we had the definition on the other page, but it was pretty confusing to write down. So think about if you can explain. You can even look at this picture and use that to help you. Okay, so non-disjunction is when the homologous chromosomes do not pull apart equally. So homologous chromosomes should pull apart equally to have an equal amount in every cell. But in non-disjunction, the homologous chromosomes do not pull apart equally, and you get too many in one cell and too few in another. So I'm going to repeat it again. Make sure you have something written down that explains non-disjunction. It's when the homologous chromosomes, when they pull apart from each other, an extra piece gets taken over and less is taken over to the other cell. So in the end, you have one cell with too many and one cell with not enough. Okay. Now, let's look at some examples, okay? Where it says write an example of a disorder, we're not going to write this one here. This example actually goes along with this picture right here. So this picture that's on your paper is the same as this picture up here, okay? So what this is showing us is that there should be a pair here, but there are 3 instead of 2. So this would be a trisomy. So we have a trisomy scenario where we have 47 chromosomes instead of 46. And if we counted these out, the way they do a chromosome chart is kind of biggest to smallest. So they go pair 1, pair 2, pair 3, pair 4. On the 21st pair that we should have, there are 3 here instead of 2. So that's why Down syndrome is known as trisomy 21. Okay, so where it says type of mutation that is shown, okay, we would go ahead and write down this is non-disjunction. So this is non-disjunction. And then when it says what's happening here, what's caused the problem, we'll say extra 21 chromosome. And then for our example, you guys can write down example of non-disjunction is also known as Turner syndrome. So in this example, there should be another pair of chromosomes here, and there is not. It's missing one, so we know it's going to be a monosomy. But this one, the last one, is the sex chromosome where you have an X and a Y if you're a boy and an X and an X if you're a girl. This is just one X, so this will be a girl, but she will not develop fully during puberty because she is missing part of that sex chromosome. So Turner syndrome, where it says write an example, Turner syndrome. And what the problem is it is missing a sex chromosome. Okay, and then I am going to let you guys make sure you have that done. And I'm going to start you off with what causes mutations in the next video.